Wonderful. Since we are kind of 12 minutes late, I will not going to introduce you, Daniela, if you um, choose to, please do, and otherwise just go on to your presentation. So without further ado, please, floor is yours.
So, hi, I'm Daniela Valdes. I'm a postdoc at the Rinaldi Ramos Lab. I work on magnetic particle imaging and other magnetic nanoparticle applications such as magnetic hyperthermia. And today for my webinar, I chose to talk about quantitative magnetic particle imaging, in particular in biomedically relevant conditions, which we do using anatomically correct phantoms. So just to start, a brief introduction. Um, as you may know, there is a necessity to be able to quantify small amounts of particles being injected into mice or subjects for treatments. And um, in different reviews, meta-analysis, we see that less than 1% of the injected dose is currently reaching solid tumors. And it doesn't matter all the efforts that are being put into changing the coding, changing the materials. Um, all the reports show that a median of 0.7% of the injected doses reaching that. And one problem that the authors of this review that I'm showing here pointed out is that only a very small amount of the reports contain more than 3 time points for imaging, and not all of this is quantitative. It's mostly qualitative data. Um, so they ask all of us for thematic analysis and to develop techniques and tools to be able to do quantitative analysis. And that's where magnetic particle imaging comes in. So you may say, why not other imaging techniques or other types of assays, such as ex vivo? Um, so when we talk about ex vivo, the problem is that it's not longitudinal. It's just an endpoint uh, analysis that we can do. And when we do imaging for um, things like treatments with magnetic particles, mostly with iron oxide, um, the problem with ex vivo is there can be iron already present in the organs, endogenous iron, and that's a limitation too, because the amount we inject might be smaller than that endogenous iron amount. Another technique quite popular is the in vivo imaging system modality, IVIS. And so doing fluorescence imaging, um, we can have problems such as tissue attenuation, chlorophyll quenching, or dequenching that can affect how we um, perceive the fluorescence signal. And there has been reports with um, showing very small amounts of particles in organs such as the liver, and then showing accumulation in tumor, but then contradicting analytical methods, which can be due to the different tissues. Then with more conventional and large-scale techniques, more established for the clinic like MRI or PET, with MRI, we have something that's semi-quantitative, and the contrast is indirect. So we probably need to add some other contrast agent, or we need a custom sequence um, to be able to quantify the particles. And with PET, it's longitudinal, it's quantitative, it's depth independent. The only thing that we can mention is we need a radioactive tracer. Of course, we need the radioactive label to stayed together with our agent for the treatment. So if we have the radioactive tracer connected to a magnetic particle, the percent um, of the label particles is gonna matter, of course, because we're actually imaging just the radioactive tracer part. And the short half-lives that these tracers can have limit us in the time points we're going to choose and how long we can keep imaging the same subject. So a way of tackling these problems is with magnetic particle imaging. In this case, I'm showing an in vivo image of a mouse that has been injected with particles 1 hour after injection, together with the CT of that mouse, and then the 3D volume rendering that contains a skeleton segmentation that we produce from the CT registered to the MPI volume. So this means that as a magnetic particle, it's just capturing the signal from the magnetic moment in an alternate magnetic field. We're gonna have no problems with the tissue attenuation, no problems with the depth. We can do longitudinal studies, we can do in vivo, but we need an anatomical technique to be able to know where the signals come from. And that's key because if we want to do, for example, quantitative analysis in a way that we're distinguishing the amounts in between organs, we need this anatomical reference. So, of course, not everything is so positive, and there are challenges with MPI signal quantification. And here I'm showing an example. This is a very good paper on challenges that we encounter when trying to quantify and even less than quantified when trying to segment organs in vivo. So this is the image of a mouse with particles injected, a few fiducials, and when we do some kind of threshold segmentation, we start seeing these shapes. So it's almost impossible to select, for example, the heart signal and the liver signal or separate between them. And another very time-consuming method that we can implement is trying to cut the segmentations manually or freehand the segmentations to kind of have organ specific ones, which of course for a lot of time points, a lot of subjects, as in an animal experiment, is very time consuming, and it's not it's quite arbitrary when the user tries to decide where to cut each organ, because of course there is a blur uh, from the um, main signal towards the background, signal decaying to the background, and towards other organs. So this problem of the leakage of signal from some region to another, it's it's well reported, and the first paper to characterize the effect and give it a name is this paper here on the right from the group of Shefoti that took the term of spillover and also shine through, because it's referring to the same phenomenon, already reported for MRI and PET, and showed how it was affecting MPI. So in this figure, they show on a live mouse that has been injected with particles with the liver saturated already, and some fiducials they're placing in different regions. They're showing how the closer they get to the liver, they're getting more and more overestimation for the signal in the fiducials. So the signal in the area we want to quantify, for example, the fiducial, is going to be affected by signal decaying from an off-target site, such as the liver. And this is a problem for all nanomedicine applications because as we know, the iron oxide particles tend to accumulate in liver and spleen. So there is a need to develop methods to do organ-specific segmentations in a non-arbitrary way, and then account for the spillover signal, counteract this overestimation in some way. So why a phantom? The phantom has many benefits. First, we can evaluate, localize, and spread out signals. We can do anatomically correct regions and mimic biomedically relevant scenarios. But more importantly, we can consistently position the tracer in the cavity, and we can validate against the ground truth iron mass we're placing in the cavity. So just to show you a little bit, all the results here are from a paper that's currently under review. This is the mouse phantom design that we're using, and the bed we're using to place the phantom. And you may see we have two brain ports to place capillaries on it, two lung ports, one liver cavity, and one flank cavity. And this is how the phantom looks here on the right. When we print it with clear resin, you can distinguish the empty liver cavity here and the holes for the brain lung ports. This is a model that doesn't have the flank. We can, as the phantom is modular, we can change this bottom part with one that has an attachment for the flank, and we can vary also the volumes for the flank. So the experimental design is basically using this phantom coming from the DC Mouse Atlas. So the dimensions are those of a mouse, and the cavities are placed in the anatomically correct positions. And we're going to analyze signals in the brain, the lung, and the flank in the presence of a very strong liver signal. So mimicking the accumulations we see in vivo. For that, we chose to fill the liver cavity with 100 micrograms of iron and then have different test masses in either brain, lung, or flank. And these are the masses we chose. And of course, we're really interested in this lower range, which is below 1% injected dose, which is what we are currently encountering in solid tumors. Why brain, lung, and flank? So, and why capillaries and cavities? So the brain and lung are going to be more localized tumors. The brain is, the brain and lung are internal tumors, localized since they're going to be capillaries. And then the flank, it's a more spread out tumor and more superficial, mimicking when we inject particles into the flank of a mouse, and the ones that we culture in vitro and to study tumor models. The MPI scans we did were 2D and 3D. And first, I'm going to show the results from the conventional technique, the threshold, in particular, the 50% threshold. So that means we're going to select the region of interest, in this case, the brain. I'm showing here. Search for the maximum signal there. Threshold from that maximum value to 50% of that value. And then we're going to have this contour marking our segmentation. So a little note is all the data I'm going to show you of the scans is going to be logarithmically transformed, just for visualization purposes because we have a large dynamic range in the images. Here on the left, you can see the um, scan, a 2D scan for the test mass 2.5 micrograms of iron in the brain, and then the liver we're using for every single scan with 100 micrograms of iron. And it's very difficult to see the signal in the brain, although this is the largest mass we're testing. So we grab the scan and we apply the logarithm to every single voxel, and this is how it looks. So this is just for visualization. All segmentations and quantifications were done in the original non-transformed form. Just to start with, for 1 microgram in the region of interest and a 2D scan, we can compare the different regions and kind of see how they're different. I said that the brain was more localized than the flank, which is more spread out. And the lung here is, oh, sorry. The lung here is also localized, but you might notice that the highest values of signal are in the lung, ROI, then the brain, then the flank. Why is that? The lung is very, very close to the liver. So the lung data set is going to serve us to characterize the spillover from the liver to the lung and see this overestimation that I told you about. The brain is far away from the liver, pretty localized, high SNR, so we should have no problems with this. But the flank is a case where it is far away from the liver, but the signal values are lower. The SNR is lower too, and it's more spread out. So for the same mass, if we compare brain and flank, flank is going to have lower signal values, and it's going to be more difficult to distinguish from the background, as you see here. So let's start with the brain. We know the brain is the easy one. So I'm showing the limit of the range we tested, the smallest mass and the largest mass for both 2D and 3D. You see in 3D, there are no problems when trying to segment with the threshold. Neither we don't have any problems with the 2D in the larger mass. But as we go down, we start encountering this problem where we can actually find the maximum, but when we select the voxels that contain 50% of that maximum, we have something that's connected to a halo around the liver. This means signal is decaying from the liver, since this has this symmetry. And the values of the maximum in this case are so low that are connecting with parts of the background and also from signal decaying from the liver. A little far away, but still. So this means that in the lower part of the range, we're testing for 2D. We cannot segment. That means we cannot quantify because this is not the region of interest we want to quantify. So here in the parity plot for quantified iron mass and nominal iron mass, you might see that for 2D, we're missing the last two points where this problem is happening. But for 3D, the quantification is pretty good. We don't have problems segmenting. For 2D, it's not that bad since we consider we are actually quantifying successfully 3 of the masses below 1% injected dose. So for small localized signals, for example, in the brain of a mouse, we can quantify below 1% of the injected dose. Of course, the smaller the masses get, the more challenging it is. But It's great. What happens with the flank? So when we start seeing the flank, 2D and 3D volumes, for the largest mass we tested, we have no problems in segmenting, but as we go down, we start seeing these segmentations that are connected with some other parts, like either the background for 2D here and the spillover from the liver and some background for the 3D. Here you're not able to see it since this is a slice of the 3D volume, but these two bits are connected in 3D. So yeah, they're connected from the flank to this halo by some parts of the background. So the user cannot go ahead and cut the spillover signal from the liver that's connected to the flank um, in a non-arbitrary way since they are connected. What happens when we quantify the ones that we can actually distinguish as the flank? For 3D, it's a little bit more noisy than for the localized brain signals. Um, and we could not quantify the lower one because we have this connected signal problem. And for 2D, we could quantify some of them, but there is clearly an overestimation. So this can be because we could distinguish the signal as being in the flank, but as the signal levels are lower than those in the brain, since it's a larger volume, we're taking parts of the background into our segmentation and not noticing it because they're close to it. And also, we can have some influence contributions from the background in the places where the signal was, but the signal is low, so that might contribute. So low SNR, we have this overestimation. So of course, more spread out signals, similar distance from the liver as in the brain, but it's more challenging. What happens with the lung? This is a very challenging case. We can see, we can quantify only 4 of the test masses, and we have this spurious signal connecting to the lung region, and sometimes we cannot even distinguish the lung signal, for example, in 2D in the smallest depth mass. So when we look at the parity plot, these are the 4 points we could actually segment and quantify, and none of these are 2D. Since even for the largest mass, we have this spurious signal connecting to the lung region. So of course, high off-target accumulation is affecting or region of interest quantification, and we need some way to prevent this. So this is where we tested another method. So we grab the volume from a thresholded segmentation, in particular the 1 micrograms of iron, since it was the one we wanted to correctly quantify. We grabbed that volume as a reference and created these spheres with that volume around the maximum. This means that sometimes when we couldn't, for example, quantify, even we couldn't segment, if we could find the maximum, as in this case, when we place a sphere around it, we're cutting all this spurious signal out of the segmentation, and we have a way of making the segmentation possible. So when we start looking at the data, we have from left to right, brain data set, flank data set, and lung. The brain is almost not affected by this new segmentation method. We can still quantify both in 2D, 3D. Of course, the one that we couldn't quantify in 2D because we had the background connected to the region, now we can. And for the flank and the lung, we can cut the background out of the segmentation from the flank and the spillover from the liver in the lung case. But we start seeing, especially for 2D, this offset we have. Um, it's more clear in the lung, and that means there's some contribution to our quantified iron mass, which can be, since it's very small, for the flank case, it can be the background contributing to the signal in the ear. And in the lung, it's more accentuated, so it's the signal that's decaying from the high accumulation in the liver. So the use of a constant volume segment as a sphere, for example, can enable us to do segmentations in challenging cases, but for low SNR scans as in the flank or cases that are highly affected from the spillover, as in the lung, we still have some problems. So I wanted to take you back to this comparison, and all these offsets we have are clear in these images, since for the flank, you kind of see the signal connecting with the background, not as in the brain. Remember, this is the same destinus placed in different regions. And for the lung, although it's the same capillary that we use for the brain, you see a higher value for signals in this region than in the brain, because the contribution from the liver is summing up with the contributions that are actually coming from the capillary in the lung. So finally, how do we solve this? So I developed the subtraction method. So it consists of grabbing the subject scan, the ones that we were analyzing, and then acquiring a reference scan that it's the same phantom with just the liver field and no capillary or cavity filled in for the region of interest, and then digitally subtracting the signal from the reference scan. This means that if in this sphere we have signal coming from the background or the spillover from the liver, we're gonna subtract that contribution from the subject scan. A little note, side note here. I'm talking about quantifying from capillaries and cavities. And of course, this cannot be done with the same reference sample. Our lab has this paper that characterizes this phenomenon. The difference we get from the sample and reference sample volumes in quantification. And you can see that when you try to quantify large volumes with small volume reference sample, as you increase the volume of the sample you're quantifying, the error increases. And when you use a larger reference sample, of course, is the contrary. As the volume of the sample you're quantifying is smaller, the error increases. So this means if you want to quantify a signal coming from a very localized spot, you need reference samples for your calibration curve that have similar volumes. And when you are trying to quantify more spread out signals, like in the flank, you need a volume that it's similar to that cavity. So this is why I'm showing you that for brain and lung, we use a calibration curve that we acquire for capillaries, and then to quantify the flank signal, we filled out cavities with the same volume and grabbed the calibration curve from those samples to quantify the flank cavity. So coming back to the subtraction strategy, we see that for the brain, it's not affected. We can see maybe the 2D improving a little bit, being closer to the predicted values. But when we look at the flank and lung, we see that this effect that we had for 2D, where we had the offset, is no longer present. And we have a better agreement between what we quantify and the ground truth. We can notice that the small values for 2D, the small test masses, can sometimes come up as negative. So our limit of quantification is around 0.25% of the injected dose for these cases in 2D. And it gets even better for 3D. For 3D, we're quantifying around 0.25% injected dose for the flank, so for a spread out signal, but we're reaching 0.05% for the lung, for the capillary signals. So the subtraction of the spillover coming from accumulation in off-target sites or the background enhances the limit of quantification. And this is a strategy we can try to translate into in vivo imaging. So I hope I convince you that threshold-based quantification is reliable when we are quantifying isolated high concentration signals, but it's more challenging when we are dealing with lower SNR or images that are affected by spillover. So in those cases, a subtraction strategy and correct segmentation are really important if we want to have a quantitative analysis. And this is really important for in vivo imaging, since we'll always have high liver and spleen accumulation when doing a systemical injection. So I want to thank my team for these for this work, and also all of the people in the Rinaldo Ramos lab, and acknowledge the funding that made this work possible. And with that, I have
Open
time
for
for some
questions, I suppose. Yes. Thank you so much for your presentation, and well within the time. Thank you. Thank you so much. Very inspiring, the whole group, basically, and your work as well. Do we have any questions from the audience? While we're waiting, you could type your question or you could raise your hand and ask your question. I don't see anything happening so far. It is, of course, as I'm not sure, with one of the first slides you had basically your phantom, and it's still under review. So it's I suppose the work you're just starting with. Could you take us a little bit through validation and ground truth? In a way, how do you establish, how do you envision to establish ground truth for iron quantification in your model or models, if you're going to extend? And how well, in your opinion, because I suppose you started from, how well does it translate to in vivo conditions? Because you do have experience with that as well, more than anyone else. So if you could take us through that.
Yeah. Okay. So I don't know if the ground truth question was referring to the phantom. exclusively, or you're thinking
Both,
of?
basically,
Okay,
both.
okay,
Yes.
yeah. Okay, so for these work, since we have the phantom, it's really easy to just what I'm doing is grabbing the stock in which I determine the iron concentration through the phenanthrene assay. And then I'm preparing the samples through dilution series. And I'm preparing the capillaries and the flank cavities like that. So I know how much I put into the capillary and the cavities. I'm filling the capillaries with a volume of 2.5 microliters. They're not completely filled out. We have a capillary filling guide to make sure that um, the signal is inside the phantom each time you prepare a capillary. So this filling guide will have um, a mark for brain and lung, for example, and so you fill the capillary and you prevent the solution uh, to be higher than that mark in the filling guide, so that it's that's telling you that the signal is going to be inside the phantom when you image. And then for the cavity, it's pretty easy because it has like a spherical cavity. It has a port to fill and an air vent to be able to fill it in a way that have the you don't have bubbles inside. So basically, as you fill it, the air is coming out of this air vent. Um, that's how we are preparing the phantom configurations. And then, uh, of course, I'm doing this thing that I show you. So I'm collecting calibration curves for 2D and 3D for each type of sample I'm preparing. So I have a 2D and 3D image of different capillaries with the same volumes that I'm using for the phantom scans. And then the same thing for the cavity, which is 65 microliters. And then, of course, you'll have a calibration curve for threshold, a calibration curve for the constant volume for each type of image and each type of sample. So a lot of calibration curves. Um,
Useful.
and then you'll have to quantify each type of scan and each type of sample with the corresponding calibration curve. For in vivo, it's more challenging, of course, because at first when you inject and you have accumulation, you don't really know the volume uh, of the organs you want to quantify. Um, when you're analyzing the images, you can estimate a volume. Uh, and then you can prepare a calibration curve with a similar volume, um, referring, of course, to this paper, which talks about how the sample and the uh, reference samples have to be similar in volume to be able to obtain a good agreement with the ground truth value.
Are you,
And
I'm sorry to interrupt you here.
yeah,
Are you suggesting we have some kind of dynamic segment threshold? So you segment, you have a volume. Let's assume the volume is one centimeter cube to stay in the metric system. So we
yeah.
have 1 centimeter cube. It's your target volume for liver, for example, and then you have a series of thresholds. Let's say you start from zero point 0.4 and you go up till you have a segmentation of 1 centimeter cube, and then you assume that would be your liver.
Now, I'm more talking about imagine you have the liver and
Mm.
you want to do threshold. let's say you want to do threshold to keep it simple, and you have 1 centimeter cube as the volume,
The target
you can
volume,
see,
yes. Mm.
yeah, um, the actual volume. Like you can grab the CT and segment it. Um, so you know that once you segment the MPI image, to quantify that, you will need a calibration curve constructed with samples that have around 1 centimeter cube in volume. So this we
Right.
do by preparing through 3D printing, as we do with the mouse. We prepare cavities with different volumes, and we grab the cavities that kind of match the
I
volume.
get that. I get it now. Thank you.
Yeah.
Thank you. I get it now. But that is somehow you could do that post scan as well. So you could do your
Yes.
MRI/CT. Use
Yeah.
let's concentrate on the liver because it's big. It's easy to talk about it. Very
Yes.
easier than spleen or something else. So you go in. You take your CT. You segment your liver, and you know the volume, the target volume for the liver,
Yeah.
and then for that specific volume, you could basically make your curves, right? So you have your
Yes.
reference and right. Thank you.
Of
Thank you
of
for
course,
clarifying that.
of course, there are other reports that use capillaries to quantify larger volumes or cavities, not really taking into account the volume. That's gonna have some error, as this paper shows, but
It's
it's a starting point.
it's a
Um,
starting point. It's
yeah,
going to have to develop in a certain direction. Which direction we don't know yet. But from
yeah,
your point of view, Um, which direction, I suppose yours, but what do you see as the most accurate and robust method to do this? Because it's not trivial. In whatever you do, it's not trivial, basically.
yeah, yeah. It's not trivial, and if you want to quantify different organs, you need a very large amount of samples prepared for your
Yes,
calibration curve
it's
because
time
the
consuming.
liver,
Yes.
yeah, it's really time consuming. So yeah, by now I'm doing this. I'm distinguishing between localized and spread out signals and trying to um, make sure the volumes match.
Mhm.
But of course, for in vivo, depending on what you want to quantify, it can take a lot of samples to prepare.
It can take a lot of sample. It's time consuming. It's
Yeah,
expensive on materials.
yeah.
What would
So
be your envisioned application that would warrant basically the use of capacity in terms of people, but also money in terms of scans and material used, etc. Because as far as I could see from my side, it is extremely time consuming and to reach a certain accuracy, and it's expensive because it requires a lot of materials. And the question is, do we actually need to go that far and be that accurate and robust? What's your vision on that?
so I think for the time consuming part, the the important thing would be having an kind of like somehow automated workflow
Mm.
that doesn't um, take too much time and
On a
segmentation,
processing, right?
yeah, on the
Mm.
processing and a method that's not very difficult for the segmentations because
Mm.
here I'm doing spheres, constant volume spheres, which are not super complex. It's when you can find the maximum, it's pretty easy just to construct the spherical segmentation. And I think keeping the segmentation simple, it's key because you have to segment all the in vivo scans for all the time points and all the reference samples in the same way. So if it's overly complicated, it cannot be done.
Yeah, it takes away robustness. More you complicate, less robust the procedure is. Thank you. There is actually a question, and it's
Okay.
very much connecting to what we just discussed. There, Lorenzi asked, well, thank you for the presentation. Would like to ask you about the software or tool used for the ROI segmentation.
Okay. So for all of the for the work that I showed you, we're using 3Slicer to load the images and um, basically analyze it. And I'm constructing Python script with the Slicer API, and I run the scripts in Slicer. So I have a workflow that I set up that it's mostly this that I show here. So with my script, I load the image, I register the CT to the MPI, and I segment the body of the phantom, like the body of the mouse. So that will get you the first image on the left, which you have the MPI and the contour of the phantom. Um, that's registered. Uh, then my script allows the user to select like a rectangular region around the area you want to quantify. So if you're loading brain, it will look something like that, like the ROI segmentation image here. So the user will have to say, okay, anatomically, um, the region I want to quantify is around this area, and the script then will search for the maximum in that ROI region, that ROI that the user selected. And once you have the maximum, it will create a segmentation with the criteria you are actually applying. In this case, I'm showing threshold, but it's the same thing with sphere. Actually, my script does all of them together. It finds the maximum and then creates the threshold segmentation and then places a sphere around it. Um, and that's the same thing for the reference scan. And then you have the script has a kind of like an interactive part where the user can intervene. For example, if the threshold had two different regions, one around the ROI and another one grabbing some values from the background, you can crop the ones that are not connected to the ROI. And if not, you are not able to threshold, which is this case that I showed. Like you can crop this part on the bottom of the mouse from
Mm.
the thresholding image to segment cropping image because they're not connected, but this scale is connected, so you cannot crop it. So that's kind of like the workflow that I created with the script. Um, and that's what I was referring to. You have to have some kind of script to be able to analyze all the images the same way, and prevent like
Yeah.
arbitrariness, but also to have some way of interacting with it, and like this last part that I told you, and to have the human intervention. Like I want to
The,
quantify the
the
brain,
the pruning
so
afterwards.
yeah, I want to quantify the brain. So if this is not connected to the brain, I'm just cropping this, and yeah, so you have a little bit of both worlds, not
I've
completely automated.
Yeah, I don't see it completely automated just yet. There's a lot of things need happening before we do that.
Yeah.
I do have one or two follow-up questions on that. I suppose the because you're referring to image registration between CT and MPI.
Yes.
That's also happening in 3D slicer or based on fiducial markers, correct?
Okay, so for registration, what we do is we create a registration dataset consisting of four fiducials placed. Two of them are placed on the bed, coplanar, and two are outside that plane, vertical, um, on the sides of the bed, and are at different heights. So that way we we're trying to cover the whole bed to reduce the error um, throughout the field of view. So if you had all the fiducials like center on the bed, or when you um, have your transformation, the error will be larger when you get away from that region where you place the fiducials. So in this case, we're trying to cover the whole field of view with these 4 fiducials placed in different planes. And then we load these images into 3D Slicer, both the CT of the fiducial data set and the MPI and the MPI. And then we use the landmark registration tool, which requires the user to select the placement or of the fiducial
In both
markers
images.
in both images.
Right.
And then you'll have the transformation matrix to apply to all your study images.
Yep.
Yeah.
Clear
Okay.
on that. Let me see. Just a quick question, Lorenzi, if I'm pronouncing your name correctly. Does this clarify, or do you have any follow-up questions?
Yeah, the pronunciation is perfect, and also thank you, Daniela, for the answer. It was complete. Thank you very much.
Thank you.
Anyone else on more questions? In the meantime, I just a quick maybe remark rather than a question from the images you're showing here, and you discussed really in details what happens when you basically accidentally you connect your in this case part in the brain to other parts in an image. That gives a bit away that you are using a global thresholding. And what you could do, you could choose to take actually, because you already selected your ROI in the brain, like a cubical small region. You could basically say, search for Smax over there and only threshold within that cubicle, and do
Yes.
not bother with anything else. Is there a reason you're not choosing for that?
Well, we do that sometimes, especially for in vivo, since the signal connecting between organs is
There's a
more
lot of
pronounced than
bleeding.
in this.
Yes.
Yeah, it's more pronounced than in these images because you have the circulation and you have a lot of vasculature at short time points. So there, if you want to segment something, you really need to crop out a spurious signal. Here I'm just testing the methods in a more like systemically applied workflow to all the images. So yeah, there are
I
things
understand that. Yes.
I could, for example, cut around the ROI, but that would introduce the user arbitrary selection from the ROI, which
It
happens
still
in
does.
every image.
It still
Yes.
does, because it picks up your S max based on your ROI
Yes,
cubicle.
but you can actually look, like you have the register images. I'm not showing here the CT completely, but you can actually see where the capillary is on the CT, and you're sure that that capillary is in the ROI that you selected. But if you leave that rectangle larger in some scan and smaller in some other, and you crop out, and basically the threshold expands to all the rectangle, then you'll have like differences in what the selection was.
You could
Because
even you could even choose and say, I have my cubicle at certain I don't know,
yeah, like a
80% of the
certain,
width. Yeah, to keep that same
yeah, like a
between the
criteria.
like exactly
Yeah.
because I get that you are searching for, in an ideal situation, you will have a script, you will load 2 images, and it will spit out the data, the Excel sheets, numbers, whatever. There's no
Yeah.
interaction whatsoever, and you're trying to
Also,
find that.
Another thing that we could do, and some papers from our lab have that. If you know that there's a lot of bleeding, you can use a higher percentage for the threshold, like 70, 80. So the important thing is to have the same criteria for all the analysis, all the images, and for your calibration curve. But yeah, then it becomes what the best method is going to be for your image, which in the in vivo imaging, it's probably going to be cropping out spurious signal, because then you end up with, if you don't crop, you end up with something like this paper was showing. Like you cannot distinguish any. Yeah.
Clear. Well, thank you for
Thank you so much.
for explanation. Just a quick recap. Does anyone have any more questions for Daniela before we close? Because we will be closing the presentation in a second. It doesn't seem, I can't see anyone raising a hand or typing a question. So Daniela, thank you so much for your
Thank you for the
presentation.
insightful questions too. Yeah,
And
really interesting to discuss.
Oh, definitely. You're coming to Portugal.
Oh no, I'm not
Oh,
coming.
you're not.
Super
You're not.
Super is going from our lab.
Super is right.
Yeah.
Okay. Well, thank you for your
Yeah,
presentation.
thank you.
Thank you for everyone
Thank you so
attending this
much.
presentation and the questions. This recording will be basically on the website nexMPI.eu to be probably to be watched and downloaded as well. It usually takes us 1 or 2 weeks before we are able to upload there to process everything, but it will be on the website. So if interested or you want to share it, just share a link to nexMPI.eu. And for everyone that is joining in Portugal, looking forward, and otherwise reach out with the questions, I suppose, to Daniela and nexMPI, whoever you need to ask any questions for. So for this moment, thank you so much. Have a nice evening from the Netherlands, and morning and afternoon to everyone else from other spots on the globe. So thank you so much. Take care. See you next time after the summer. Bye bye.